Well, hello again. Today we're going to spend some time talking about development of the cardiovascular system. I have included here your learning objectives. Again, remember you're studying out of and you should be reading out of The Developing Human by Moore, and it should be the 11th edition. Pay attention to these because your test questions will come specifically from these learning objectives. Our outline today and what we're going to do today is really talk first about the formation of the heart by two endocardial heart tubes. We're then going to talk about how that heart tube, those two heart tubes come into form one heart tube, which then begins to fold and flex, which we call bulboventricular folding. We're going to talk about forming some cushions called endocardial cushions, and these are very important structures. Then we'll talk on 4 different sessions on in our time this morning on AV canal septation, atrial septation, septation of the ventricle, and then septation of the outflow channels or conal truncal septation. To get started, the first thing we need to do is acquaint ourselves with the major embryologic blood vessels that we're going to be talking about. So first up here in red are the vitelline vessels. Those are going to develop down here. Here's vitelline vein, vitelline artery. Those are going to develop down here in the umbilical vesicle or yolk sac. They're derived from splanchnic mesoderm, and they will grow out and flow out into the yolk stalk and then into the embryo. Shown in green are umbilical vesicles, or vessels, not vesicles, umbilical vessels. And we find those in the connecting stalk here. The umbilical vein and the umbilical artery are going to form in the connecting stalk and connect back here to the placenta. We have in yellow the cardinal veins, anterior common and posterior cardinal veins. So shown right here is the anterior cardinal vein. This is posterior cardinal vein, and where they're joined together is the common cardinal vein. And the common cardinal vein is going to empty into this little region of the heart called the sinus venosus and enter venous blood into the venous end of the developing heart. Shown in blue here is the dorsal aorta, which is actually in red in the diagram. This will be the dorsal aorta, and the vitelline artery will empty into and connect with the dorsal aorta. And then in gray here, we see endocardial heart tubes. We're past the point of the 2 endocardial heart tubes in this diagram. They've already fused to form a single endocardial heart tube, and these are going to be formed by splanchnic mesoderm that we're going to see in just a minute. All right, so those are your players, the primitive heart, but at the rostral end of the primitive heart, where we have the outflow of the heart, this would be the atrial or the arterial end of the heart, we have these aortic arches. These are components of the pharyngeal arches and blood vessels that are important. And we're going to talk in our second session today about these guys and what are their contributions to the adult cardiovascular system. So let's begin our discussion. We have to go all the way back to week 3 of development, end of week 3, beginning of week 4. And we have to look at this developing embryo. We've taken off the amniotic sac, so we're looking down from the amniotic cavity onto the upper surface of the developing embryo. Here's the neural plate forming. There are somites, and you can see out here in the lateral mesoderm, we're getting angiogenesis occurring. And at the rostral end of this, we have this primitive heart that is beginning to develop. So if we take a sagittal section through this embryo and look at it from the side, we would see something that looks like this, where you have your oropharyngeal membrane, future mouth region. Here's your primitive heart tube beginning to develop up there. Here's the primitive brain and the notochord, and we're beginning to get our head fold. But what's important here is notice that the heart tube, and actually there's one on each side, you'll see it in a minute, 2 tubes here, they are rostral towards the head of the oropharyngeal membrane. But with this rotation, head folding rotation that you're seeing here, this heart tubes come together, they will fuse with each other, one from each side, and they will come to lie in the neck region, caudal to the oropharyngeal membrane or the future mouth region. So you've seen all this before, but we just need to put it in context for you. So let's take a cross section through here. That's what we'll see here at level B. We've taken a cross section right through here, and we're looking at it right here. You'll see the neural plate with the neural folds and the neural groove. You can see there's two dorsal aortae, one on each side, and you can see that there's an endocardial tube here and another endocardial tube here, out lateral on each side, all right? And so those endocardial tubes are there. We're going to get the head folding, and those two tubes are going to begin to fuse with each other to form a single endocardial heart tube or single heart tube, shown right there. And this is the wall of the heart tube. This lining here is endothelium, okay? Comes from, it's a derivative of endothelium, all right? Blood cells are forming inside of there. Now, we have this other tissue outside here that's called the myoepicardial mantle, and it's divided into two parts that you can see here, the pink, which will form the myocardium or the musculature of the heart. And we have this part out here, which will form the little more pink here is a myoepicardium. This is cardiac jelly. Sorry, the light pink is the cardiac jelly. Then just outside of that is the myocardium, which will form the musculature of the heart. And at the very periphery, a very thin layer of dense connective tissue, the epicardium, which will form the visceral pericardium. And then these cavities here are your pericardial cavities, which we talked about a couple of weeks ago. A little farther along in development, you can see the pericardial cavity. You can see the endothelium of the heart tube with the blood cells inside. You can see the lighter pink myocardium has proliferated now, more muscle cell, heart muscle tissue developing. And then outside of that is the epicardium, visceral pericardium, very dense connective tissue. If we take and do a sagittal section through the developing heart, now we're going to be down in the neck region, you're going to see that the heart, this heart tube, is beginning to fold on itself, forming what we call a bulboventricular loop or bulboventricular fold. This would be the venous end of the heart. This would be the arterial end of the heart. The blood's flowing in, venous blood flowing in, and going out through the truncus arteriosus to the aortic arches. This space right here is a space you see right there. It's called the transverse pericardial sinus, and it's where the dorsal mesocardium has degenerated. And you can actually, in lab, when you get into back into Gross lab, you can stick your finger behind the pulmonary trunk and the arch and the ascending aorta there through this transverse pericardial sinus that you see right there. And that pericardial sinus formed as the dorsal mesocardium or the dorsal mesentery of the heart begin to degenerate. All right, so epicardial heart tubes, dorsal mesocardium, and transverse pericardial sinus. So let's look at this heart tube. Let's blow it up. We have endothelium. We have this other stuff called cardiac jelly. It's sort of like jello sitting right outside of, outside of the endocardial heart tube. Outside of the cardiac jelly, we have myocardium coming right in here. And that myocardium is going to rapidly proliferate to form a lot of muscle. And at the very outside here, this little black line right there, we have the epicardium or visceral pericardium. So you need to pay attention to those issues. Myoepicardial mantle would be everything except the endothelium. Cardiac jelly would be in here in the lighter pink. Then we would have the myocardium, which forms the muscles of the heart, and then the epicardium on the outside. So Let's go back a little ways and let's start with these two endocardial heart tubes and see how that inside the pericardial cavity, they begin to fuse with each other and eventually give rise to this single heart tube that we saw. And as we get down here to diagram D, you can see that this heart tube is beginning to fold on itself, all right? And that fold is called the bulboventricular fold, right in here and right there, that bulboventricular fold. But we now have some specific names to this heart tube. The first thing we want to look at is called the truncus arteriosus. It's the most rostral part of this heart tube, still inside the pericardial cavity. This is your pericardial cavity, all in here. So truncus arteriosus, most rostral, bulbus cordis, forming the just caudal to the truncus arteriosus. These two will form the outflow tracts of the heart. Then you have the primitive ventricle, primitive because it's one large open space inside that heart. You have the primitive atria right here, where instead of two atria, you have one atria. And then below the, or caudal to the primitive atria, you have this opening right here, this area called the sinus venosus, and you have a right and a left sinus horn emptying into the sinus venosus. So this would be the venous end of the heart, umbilical vein, and vitelline veins draining into the sinus venosus through the right and left sinus horns into the primitive atria, then to the ventricle, bulbus cordis, and out the truncus arteriosus. And here you can see this a little farther along in development. Truncus arteriosus, bulbus cordis, ventricle now has enlarged, and you can see the primitive atrium back here, and sinus horns and primitive sinus venosus right in this region. But with this bulboventricular folding that's happening here, the venous end of the heart is moving dorsally, and it's going to rise and come to set. The atria are going to come to set right up in this region. And we see that in this diagram. All right. So here's your bulbus cordis, truncus arteriosus, primitive ventricle, primitive atrium, sinus venosus right here, and sinus horns. The heart is going to rotate during this bulboventricular fold 90 degrees in the counterclockwise direction. That's viewed from above to down. So you can see my arrows here. I'm giving you that counterclockwise rotation 90 degrees. And so now the lateral surface moves to the ventral surface. The ventral moves to the left, and the left side over here would move to the right or to the back dorsally. So now we have a structure that looks like that. And remember, during all of this, day 25, the venous ends are beginning to go dorsally and rise. And if we get that 90-degree rotation, now we have the atria up here on the rostral part of the heart, but they're dorsal to the truncus arteriosus and conus cordis, which are the outflow tracts of the heart. Here's your primitive ventricle, primitive atria. Here's the pharyngeal arches that are going to give rise to structures that we'll look at in a minute. All of this happens because of bulboventricular looping. Here we see this at day 30 and at day 35. And you can just see how the heart is enlarging. And now we're having right and left atria, right and left ventricles. That makes sense? So if we take the heart we just looked at and we do a sagittal section through it, this will be ventral surface, this will be dorsal. Here's the atria, the primitive atria that's moved behind the truncus arteriosus. And you have the venous end, venous blood draining in through the sinus venosus into the primitive atria, down into the primitive ventricle, and out the common truncus arteriosus and bulbus cordis to enter the aortic arches. So that's first figure. Second figure, we have these little structures right here beginning to develop on the area of the canal between the primitive atria and primitive ventricle. And these darker pink things are called the endocardial cushions. And you're seeing a ventral endocardial cushion and a dorsal endocardial cushion. Here you can see those endocardial cushions have fused. So now you have a single atria and a ventricle, and they communicate. And if we take a section here, a coronal section through here, look at it from the front going to dorsal, you can see the fused endocardial cushions here. You can see your atria here, and you can see your ventricle here. And now you're seeing growths in the roof of the atria that are beginning to grow down to fuse with the endocardial cushions to form right and left atria. And from the floor, you see a primitive interventricular septum growing up, a muscular component that will help to separate the ventricle into right and left ventricles. In this process, particularly the process that I want to talk about, these endocardial cushions that are forming right here, this would be the dorsal, this would be the ventral endocardial cushion. We talked about cardiac jelly helping to form these endocardial cushions. But what's really happening is neural crest cells that are specifically called cardiac neural crest cells are migrating into these regions. Now remember, neural crest cells come from the neural tube, and they're neuroectoderm in origin. They are not mesoderm. So these cardiac neural crest cells, because they're ectoderm, they have adhesions and they will not migrate. They will stay adhered to each other. So they have to undergo epithelial to mesenchymal transition. They have to become mesenchymal, and they do that by the process of EMT that we talked about with lung development. And now they can migrate from the pharyngeal arches up in the neck region down into the developing heart and give rise to endocardial cushions, as well as to ridges, bulbar ridges, that are going to form and help to form the outflow tracts of the developing heart. Cardiac neural crest cells, very important. They need to develop. If they don't, you have all kinds of septal defects in the heart, okay? Very important, cardiac neural crest cells. And they undergo EMT, and slug is one of the factors that is being produced by them as they undergo EMT. So there you have that. So now let's move to atrial septation. We're moving into the 5th week of development. This is our developing heart, and we're taking a sagittal section through it. You see it in this lane. And we're taking a coronal section through it over here on this side, all right? And there's some names you need to pay attention to here. I've outlined these and I've bolded them in black. So in this first sagittal section, what we see is a membrane growing from the roof and posterior wall of the primitive atrium, and this darker pink membrane is called the septum primum, or the first primum. And it's going to grow all the way down here and eventually fuse with these endocardial cushions that you see right here. But as the septum primum grows down, this opening underneath it, which is called the foramen primum, or another name is ostium primum, you might see that, but we will try to stick with foramen primum. That's an opening that allows the right atrium here to communicate with the left atrium on the other side. Why is that? Because we have oxygenated blood here, and we needed to get it to the left atrium and down to the left ventricle and out to the rest of the body. So right atrium, right to left shunt of oxygenated blood coming from the placenta, and the septum primum is growing down and is eventually going to fuse with the endocardial cushions. But we have that opening there, that large foramen primum. So as the septum primum grows down and begins to fuse with the endocardial cushions, new openings form in the septum primum, all right? And those openings will now form the foramen secundum, the second opening. And you can see that right there. So just a hole there and a hole here. The foramen primum is closing off, and now the foramen secundum is opening. And as we go a little farther down our time in development, the septum secundum begins to form just to the right of the septum primum and begins to grow down from the roof and the ventral surface and grows down towards the base here in the endocardial cushions. So you can see that developing second secundum here. Here's the septum primum, here's a septum secundum, and that septum secundum is going to grow down this way. All right? And that hole between these guys is going to be called the foramen ovale. The foramen ovale. So here you see the septum primum has grown down and fused, and you have the foramen secundum. Okay, the second opening has developed up here. And now you see the septum secundum beginning to grow down. And actually, as it grows down, it covers over that opening of the foramen secundum. But it grows down and it doesn't completely fuse at the bottom. And so now you have blood coming in from the inferior vena cava that's relatively rich in oxygen, and it comes into the right atria. And as it comes into the right atria, that blood is going to, at early stages, go through here, through this area called the foramen ovale. But as the septum secundum comes down and crosses over, what happens to the blood here in the right atrium? Can it get to the left atrium? Well, what happens is we build up pressure in the right atrium. We push the septum primum to the left, opening the foramen ovale. So the septum primum acts as the valve of the foramen ovale with increased pressure in the right atrium, opening it so now blood can go into the left atrium. All right, very simple. Two membranes growing down, septum primum, septum secundum, and septum primum will have a little opening in it. The septum secundum will as well, and blood coming into the right atrium will push with increased right atrial pressure, push the septum primum to the left and open the foramen ovale, allowing blood to go in. So what is the valve of the foramen ovale? It would be the septum primum. So prenatally, we have right to left shunts. Before birth, the right atrial pressure is higher because of blood coming in from the inferior vena cava. Blood pressure increases, pushes the septum primum to the right, and opens the foramen ovale. After birth, the lungs are inflated. The blood vessels in the lungs can now take more blood. So we have blood going from the right ventricle into the lungs. The lungs are inflated. Now we have more blood returning into the left atrium. And the left atrium now is under high pressure, and it pushes the septum primum to the right, functionally closing the fossa ovale. And over time, adhesions will develop and we'll get an anatomical closure of the fossa ovale. And so we close that right to left shunt. So there's other right to left shunts. The first one we've just talked about is a foramen ovale. We have another one which we'll talk about in our next session a little later, called the ductus arteriosus, which connects the left pulmonary trunk with the descending aorta, and we'll talk about that, and you'll see why that's important. What we're not going to have time to talk about today is the ductus venosus, which is where the umbilical vein from the placenta carrying oxygenated blood bypasses the liver and connects to the inferior vena cava through the ductus venosus. And that's, again, a right to left shunt. We don't need the liver in utero. In utero, before birth, the functions of the liver are heavily performed by the placenta. So we can bypass the liver and just give it a little blood supply and let it continue to develop. Postnatally, that liver has to function. All right. Now, I'm not going to spend any time going over atrial septal defects, but there are a variety. There's probe patent foramen ovale, where it's large enough so you can stick a probe through the foramen ovale, and it should close after birth, but it doesn't. It's open. That's an atrial septal defect. You can have secundum types of atrial septal defects, endocardial cushion types of atrial septal defects down here, and venous types of atrial septal defects, maybe back over in here. So I'm not going to spend time talking about those. You need to pay attention to them, be aware of them. Dr. Brandt will likely address and should address atrial septal defects as well as ventricular septal defects in her session with you a little later. So the good news is septation of the ventricle is not nearly as complex as septation of the atria. So let's take a look at it. So here we are with the bulboventricular folding. Let's get our orientation. Here's the aortic arches. Here's the truncus arteriosus, bulbus cordis. Here's the common ventricle, beginning of the right and the left. Here's the atrium, sinus venosus. Here's the atrioventricular canal. Again, what do we have here? We have a sagittal section, all right, in this view. And what you can see is growing up out of the floor in the latter part of the middle to the latter part of the 4th week of development, we see this muscular growth coming out of the floor of the common ventricle. And the ventricles here are beginning to proliferate and enlarge. And we have this muscular upgrowth forming a muscular interventricular septum. However, it doesn't close up here initially and leaves a foramen here called the interventricular foramen. So we still have right to left communication of blood in utero early on. This would not be compatible with life postnatally, okay? So we need to close this interventricular foramen. And how do we do that? We do that, and now we're looking at a coronal section through the heart. You see right atria, left atria, right ventricle, left ventricle. You can see the interventricular septum here and here. But here you can see now that a membrane has formed from this interventricular septum closing that interventricular foramen. So we have two parts to our definitive ventricular septum. We have the interventricular or muscular part down here that grows here and closes everything, almost everything up except that gap at the rostral end. But now we have a membranous part that closes that gap, and now we have a right and a left ventricle. So this is the membranous part of the interventricular septum. All right. Where does that tissue come from? Well, obviously one place it comes from is right here. What's this? These are the endocardial cushions, okay? There's endocardial cushions right there. So they give some tissue that helps to form this membranous interventricular septum. But you can see these different colored structures. We get contributions to this membranous interventricular septum from endocardial cushions shown here in blue, and from these bulbar ridges that help form the outflow tracts of the heart. The right and the left bulbar ridges all contribute to form that membranous portion of the interventricular septum, which fuses with the muscular septum here to give us our right and our left ventricles and to close them off. So we want to now talk about how we separate the outflow tracts of the heart. Here we have one common outflow tract, but you know you need two. You need a pulmonary trunk going to the lungs, and you need an aorta going to the rest of the body. So how do we do this? Well, this truncus arteriosus and bulbus cordis are common open tracts, and blood coming out of the ventricle will go into the bulbus cordis, into the truncus arteriosus. But there's a spiral septum that forms in the middle of the bulbus cordis and truncus arteriosus, a spiral septum that begins to grow. And if you section these at these different levels, you can see this ridge of tissue beginning to form. And depending on where you cut it, it's at different angles because it's a spiraled septum. And a little bit later in development, those ridges have grown to the midline and have fused with each other. So now you have a pulmonary trunk and an aorta at this level. Go down a little bit to 2 here. The aorta is over here, the pulmonary trunk's here, and you go down to 3, and now the aorta is dorsal and the pulmonary trunk's ventral because of that spiraling septum. This septum is going to proliferate rapidly, become very thick, and then cells in the center of that septum are going to begin to die off. And when they die off by apoptosis, we now get the separation of a pulmonary trunk here and an ascending aorta. All of those are formed by this truncus arteriosus, bulbus cordis, and this aortical pulmonary septum that separates those two into two components. It's that simple. Don't drive yourself any more crazy. So this would be the aortical pulmonary septum right there, and right there at different places because it's spiraling. Okay. So you can see these bulbar ridges that are here and here. They go up into the truncus arteriosus. They're going to thicken, forming the aortical pulmonary septum. But again, what you need to know is part of that aortical pulmonary septum, we have those cardiac neural crest cells migrating in and setting up housekeeping to help form those bulbar ridges, to help form the endocardial cushions, all of those. So if those cardiac neural crest cells do not come in, you have all kinds of septal defects. If they do come in, but there's too many of them, they can do things like causing transposition of the great vessels, where they build up underneath the truncus arteriosus and cause it to rise and sort of shift positions, rotate on itself. And if that's the case, then you can actually get a situation called transposition of the great vessels. So Dr. Brandt, in a little bit, will have a video for you where she will discuss clinical issues. She'll discuss atrial and septal defects, and she'll also discuss transposition of the great vessels and tetralogy of Fallot, all these well-known issues of maldevelopment of the heart. So after you answer a few questions, and Dr. Ray is going to make some questions for you, I'm going to come back with a short video to talk about pharyngeal arch derivatives that contribute to the vascular system, and also the issue of coarctation of the aorta. I'll see you back here in a few minutes. Bye-bye.